In this lecture, we're going to cover microbial diseases of the skin. And this is fun because we get to revisit some of the concepts from the previous Exam 3 material. So human skin is an important part of what immune system? The innate immune system. So it's an important part of our first line of defense from pathogens, so this includes both chemical and physical barriers. As a physical barrier, our skin is composed of layers of cells that have tight junctions, and this prevents pathogens from just being able to pass right through our skin. Our topmost layer of skin is composed of a layer of dead skin cells that are constantly being shed, and this lowers the microbial load on the surface of our skin. Skin also provides chemical barriers by secreting sweat and sebum to the surface, which slightly lowers the pH on the surface of our skin, and these secretions have a special enzyme called lysozyme, which can break down or lyse bacterial cells. So fantastic, we know a lot about the skin in relation to the immune system, but for this lecture, we're going to discuss microbial diseases of the skin. So first, we'll talk a little bit more about the structure of the skin, because we'll see that different infections can occur in different areas of the skin, depending on the infectious pathogen or how that pathogen is transmitted. By different areas, I really mean different layers of the skin. And the two main layers that we'll discuss in terms of infection are the epidermis, and then right below that, the dermis. There's also this third layer called the hypodermis, and this is primarily composed of fibrous and adipose tissue. Over the next couple slides, though, we're going to talk a little bit more in depth about these two main layers, the epidermis and the dermis. First off, the epidermis, and this is the outermost layer. Below this is the dermis, below that is the hypodermis, and the epidermis is really the thinnest layer compared to those other layers. It's composed of this layer called the stratum corneum, and this is the outermost layer, or this exterior surface that's in direct contact with the environment. And this area is rich in keratinized dead skin cells. Those are the cells that are shedding off of the surface of your skin through a process called desquamation. And you shed a lot of skin. So up to 50% of the dust in your house is your dead skin cells that you have shed. And some studies even estimate it's higher, up to 80%. And then you shed about 1.5 pounds of dead skin cells each year. That's a lot of dead skin cells, and it's really important for this desquamation process to reduce the microbial load on the surface of your skin. Next up, the dermis. So the dermis lies below your epidermis, and this is a thicker layer of skin. And it is primarily composed of this connective tissue, and it's embedded with a lot of important things, like blood vessels to provide oxygen and nutrients to your skin, nerves for your tactile interactions with the environment. You also have muscles, you have hair follicles, you have different types of glands. So you have sebaceous glands and sweat glands. And it's these sweat glands and sebaceous glands that provide these secretions to the surface of your skin and release chemical barriers like lysozyme, that enzyme that can break down bacterial cells, to control microbes on the surface of your skin. Just a quick slide for review, and this goes over some of the immune system stuff as well as the anatomy of skin. So how do sweat and sebum provide protection from microbial growth? Those glands are secreting these things like chemical barriers onto the surface of our skin, like lysozyme, but also these are going to lower the pH to prevent the overgrowth of certain microbes that can't live in lower pHs, and they're also going to help increase the pliability of our skin, so our skin doesn't break open as easily. Next up, where do you think most microbes reside on our skin? We talked about these three layers, the epidermis, the dermis, and briefly the hypodermis at the beginning there. And of course, most microbes are going to be on the epidermis, this area of the skin that is exposed directly with the environment. You do have microbes, though, that live deeper down in kind of the root of your hair follicle in the dermis, but most microbes are going to be right on the surface of your skin. And then finally, how does desquamation prevent infection? Desquamation is the shedding of those dead skin cells on the surface of your skin, and it reduces the microbial load so you don't have so many microbes on the surface of your skin, and this decreases the chance of infection and allows your skin to remove and shed possible pathogens that have attached to the surface of your skin. the normal microbiota of the skin. You've heard me say this multiple times probably already throughout this course, but you're covered in microbes, and that is fantastic. This is our normal microbiota. But that being said, do you think you have the same variety of microbes living in all different areas of your body? Do you think that the microbes that live on your nose are the same microbes that live in your armpits or between your toes? No. These are all different regions of your skin, and on our bodies we have all these different regions that have different properties that allow different communities of microbes to proliferate. The major regions of skin include moist regions like our nostrils and our armpits, drier skin on our arms and legs, and then oily skin on areas of our face and on our backs. And we'll kind of talk about all of these different areas and the different communities that hang out here. So this is a busy image. I get that. But I won't be asking you to label an image like this. I just want you to see the variety of microbial communities on different parts of the body and kind of point out the most abundant type of bacteria in each of these different types of regions. So in dry regions, you have beta proteobacteria. In moist regions, you have Corynebacterium and Staphylococcus. And then in oily or sebaceous regions, you have Propionibacteria. And Propionibacteria includes bacteria like Propionibacterium acne. So what do you think that causes? That is the causative agent of acne. And this bacteria feeds on the nutrients in sebaceous matter. So that's why it hangs out in those oily regions. And don't forget, of course, that viruses and fungi are microbes too, and we also have viruses and fungi that make up our normal microbiota. Overall, though, and definitely know this one, the most common bacteria on your skin is Staphylococcus epidermidis. Everyone has this on their skin, and it's part of your normal microbiota. So, I hope you see that we have a wide variety of microbes that make up our normal microbiota. And many of these microbes are opportunistic pathogens. And recall that opportunistic pathogens are microbes that would normally not cause an infection in healthy people with a functioning immune system. But when people have weakened immune systems or they experience an upset in their normal microbiota, these microbes can take the opportunity to proliferate and lead to an infection that may cause a disease. MRSA, or methicillin-resistant Staphylococcus aureus, is a good example of such a pathogen. About 5% of the population can asymptomatically, so they don't have symptoms, they can asymptomatically carry MRSA in their nose. This can lead to an opportunistic infection in the carrier or other people with close contact to the carrier, and I'll mention MRSA again in a few slides. Also, damage or injury to the skin from things like surgical incisions, for example, can allow bacteria on the surface of the skin to get past your first line of defense and potentially cause an infection. So that would be opportunistic pathogens that are hanging out on the surface of your skin. Once they are able to get deeper past your first line of defense, or you have a weakened immune system, they can potentially cause an infection. This slide and the next slide just cover some medical terminology concerning infections and diseases of the skin. I will not be asking you about medical terminology on the test, but I know many of you either already are in the medical field or are headed that direction, so you may find it interesting. Now we're going to dive a little bit deeper into different types of skin infections based on the type of bacteria that causes that infection. So first off, we'll talk about Staphylococcus and different types of species of Staphylococcus. Most often, infections of the skin are caused by Staphylococcus epidermidis, Staphylococcus hominis, and Staphylococcus aureus. Staphylococcus aureus in particular is pretty contagious, so that means it can be transmitted from person to person, and as I mentioned, some people are chronic nasal carriers of Staphylococcus aureus, and in particular MRSA, or methicillin-resistant Staphylococcus aureus. So MRSA is taken very seriously in hospital settings because it is so contagious. It's really dangerous as a nosocomial infection for immunocompromised patients. And it's such a dangerous infection because Staphylococcus aureus has these virulence factors that make it easier for this bacteria to evade the immune system and lead to an infection. So it has things like hemolysins, staphylolysins, and leukocidins, and these are all cytotoxic. So these are enzymes that this bacteria makes that break down cells. For example, hemolysins are breaking down red blood cells, and leukocidins are breaking down white blood cells. Infections are commonly confirmed by culturing samples on special specific media like we do in the lab, and we'll see an example of this on the next slide. This is a mannitol salt agar plate, and it's useful for identifying Staphylococcus aureus. So on the plate you have Staphylococcus aureus on the left and then Staphylococcus epidermis on the right. And this plate, like it sounds, has a high salt content. So this inhibits a lot of microbial growth, but Staphylococcus, which lives on our skin, survives great under these conditions because, like, our skin has sweat, and sweat is pretty salty, so it's used to those salty conditions. Staphylococcus aureus in particular ferments mannitol, and that produces acid, which converts this color change. So it changes the agar, which starts out as kind of this reddish color, and it changes it to a yellow whenever it is fermenting mannitol in this agar. Infections with Staphylococcus aureus in particular is often associated with pyoderma, and this means pus in the skin. So they can also be described as skin infections that are purulent, and this also means that there is pus present or it's containing pus. So pus is associated with Staphylococcus aureus infections because of those virulence factors that it has. So those leukocidins that Staphylococcus aureus produces kills white blood cells, and that inhibits our immune system, and the pus that you see that builds up around wounds are all of those dead white blood cells. And depending on the depth of these skin lesions, there are different presentations. So folliculitis, right here, this is smaller lesions that involve inflamed hair follicles, and they become infected. So this can commonly occur from skin chafing or just your hair being irritated at the follicle. Folliculitis can develop further into something called furnicles, and these are tender nodules that most often involve a hair follicle and these are going to be larger and deeper and more painful. And then next there are carbuncles, and these are even deeper, and most often they're from a cluster of furnicles that are all kind of connected under the skin. And this is going to be painful, and this is also likely going to be accompanied with a fever, because your immune system is definitely aware that this is here and this is a potential problem. And these carbuncles in particular really need to be treated and drained to prevent further infection, because this could potentially reach the bloodstream and lead to a much more dangerous systemic infection. A couple more examples of Staphylococcus infections. So we have staphylococcal scalded skin syndrome and impetigo. Staphylococcal scalded skin syndrome, SSSS, so this is caused by Staphylococcus aureus. And this is from bacteria releasing exotoxins and then leading to the skin peeling. And this occurs in children, most commonly under 6 years old, and then primarily in infants, and it's treated with intravenous antibiotics. The peeling of that top layer of skin means that the protective skin layer is compromised, and this can potentially lead to systemic infections, so it is treated very robustly and immediately. And then we have impetigo, and this again is predominantly in children. It's caused by Staphylococcus aureus most often, but can also be caused by Streptococcus pyogenes. And then this is mostly going to be treated with a topical antibiotic on the area of infection. On to streptococcal infections. So we're not talking about Staphylococcus anymore. We're talking about Streptococcus. And specifically, we're going to talk about a specific species of Streptococcus called Streptococcus pyogenes. This is the most clinically relevant species. And it's the most clinically relevant because, like we saw with Staphylococcus aureus, it has very important virulence factors that let this bacteria kind of more easily cause infection and spread. So it produces enzymes that aid in transmission and cause inflammation. So like Staphylococcus aureus, it has these enzymes like hemolysins and leukocidins that can kill cells, causing inflammation and alerting the immune system. Streptococcus pyogenes can also produce a capsule. And remember that a capsule is a type of outer glycocalyx, and the capsule, being very well organized, prevents phagocytic cells from coming along and eating and consuming and breaking down this bacteria. So some common examples of streptococcal infections from Streptococcus pyogenes are cellulitis, erysipelas, and erythema nodosum. And these are all kind of a result of Streptococcus pyogenes being introduced into the skin from some kind of maybe cut or abrasion that you get on your skin, and it's an opportunistic infection. And these all commonly happen on different parts of the body, depending on what part of the body that this bacteria infected, or different layers of the skin. So cellulitis, for example, this is most commonly going to be deeper in the skin, to the dermis or the hypodermis. Erysipelas is commonly occurring on the face, and this can also potentially cause pus to form. It can be purulent. And erythema nodosum is commonly occurring on the shins. So I'm not going to ask you to differentiate between these three. I just want you to see the different types of presentations that one type of bacteria can have depending on where this infection occurs. Another very clinically relevant infection regarding Streptococcus pyogenes is necrotizing fasciitis. And this isn't only caused by Streptococcus pyogenes, it's just most commonly caused by that bacteria. It can be caused by other bacteria as well. But necrotizing fasciitis is also called flesh-eating bacterial syndrome. It is very rare, but it is also life-threatening. So this is when bacteria infects the fascia layer. And fascia is the layer of connective tissue that's connecting your muscle to your skin. So when bacteria infects the fascia layer and starts degrading that layer, now your skin does not have access to all the nutrients and blood supplies that it needs, so that tissue starts dying or necrotizing. And this, of course, needs medical treatment that is required and needs to be surgically debrided, so that dead tissue needs to be removed. It does require many rounds of antibiotics, and amputation is necessary when this has really escalated and too much tissue loss has happened. And this can happen really quickly. This infection can potentially spread really quickly, and you can see that it is life-threatening because of how many layers deep this is happening, so it can easily reach your circulatory system and become a systemic infection. Now on to the genus Pseudomonas, and the most clinically relevant species we are interested in is Pseudomonas aeruginosa. And like we've seen with Staphylococcus aureus and Streptococcus pyogenes, this is also a common opportunistic infection. So this bacteria can just be hanging out on your skin, and given the opportunity to pass that first line of defense, it can lead to an infection. And commonly, so it's going to be an infection of wounds from damaging that first line of defense, and it's also very common for burn victims. And it can also cause infections of the ear, so otitis externa, swimmer's ear, the external part of the ear. And it's interesting because these wounds have a distinctive odor, and this is from certain virulence factors and communication molecules that this bacteria is secreting into the environment. And often people say this has a distinctive odor of grape soda or corn tortillas. I know those are very different smells. If you've ever smelled it, let me know if that's relevant and that is accurate. But it does have that distinctive odor, and that is one way that it is kind of diagnosed, is people removing the dressing from wounds and cleaning wounds and then recognizing this odor and immediately being like, oh, this may be a Pseudomonas infection, and then a culture is made to distinctly identify that infection. Another way it can be identified is that it can commonly form this kind of bluish-green pus as well from the secretion of different colored enzymes and communication enzymes that this bacteria is secreting. Pseudomonas aeruginosa does have important virulence factors that allows it to infect us. So like we've seen over and over again, these virulence factors like hemolysins to break down red blood cells, adherence factors to help Pseudomonas attach to tissues and lead to the infection, and then also secreting these different types of exotoxins that do cause tissue necrosis. So Pseudomonas aeruginosa can also cause necrotizing fasciitis like we just saw. And then it has a slime layer on the outside, so it doesn't have a capsule, but this can also help with adherence. And Pseudomonas aeruginosa tends to be resistant to lots of different types of antibiotics. So it's known to produce beta-lactamases, so penicillin is not effective against Pseudomonas. And it can also alter the porins and pumps in order to regulate the drugs that are getting into the cell. And then finally, Pseudomonas is also really good at forming biofilms and forming a community and a very resilient matrix that makes it very resistant to these drug types as well. So it is commonly treated with anti-biofilm agents that help prevent Pseudomonas aeruginosa from communicating with other bacterial cells and forming a biofilm. Now, finally, the last thing we'll talk about in terms of bacterial infections of the skin, we'll talk about anthrax. And anthrax is caused by an infection with the bacteria Bacillus anthracis. And most commonly, I think, when we think of anthrax, we think of inhalation anthrax, and we think of anthrax being used as a bioweapon. But anthrax is most commonly going to be cutaneous anthrax, or infections of the skin. And it's not nearly as deadly as inhalation anthrax. Inhalation anthrax has a much higher mortality rate compared to cutaneous anthrax. If you get cutaneous anthrax and you get an antibiotic treatment, most of the time, you're going to be perfectly fine. But specifically for cutaneous anthrax, this is a zoonotic disease most commonly because Bacillus anthracis forms endospores. It forms endospores that hang out in the soil. And so in the soil, we have these farm animals and different types of herd animals that are also hanging out in the soil. And then they can pick up these endospores from the soil. And then farmers or workers that are working with those animals or the wool or hide of those animals can come into contact with those endospores if they have some kind of cut or abrasion in their skin. And that's how these endospores are introduced into the skin. And again, most commonly, it's going to be cutaneous anthrax is the form of anthrax that people are being infected with, and that's the disease presentation. And then at the site of infection, it's going to kind of cause these sores on the skin that turn black. And you'll see here, I wanted to point out this plate here because this is a blood agar plate. And we've talked about hemolysis kind of for the previous types of bacterial cells or bacterial infections that we've talked about. But you'll see on this blood agar plate, it has this kind of grayish growth and kind of this kind of crystallized growth that's common for Bacillus anthracis, but this blood agar is not broken down. So Bacillus anthracis does not have those hemolysins. It is not beta hemolytic. It does not break down red blood cells. So it doesn't have the same virulence factors that we've seen so far. And again, I wanted to talk about this because we think of anthrax as inhalation anthrax, but cutaneous anthrax is very common and easily treatable with antibiotics. Now we are moving away from bacterial infections of the skin, and we're going to talk about an example of a viral infection of the skin. Because remember, our skin is colonized by viruses too. Viruses are also microbes, don't forget about them. And then some examples of viruses that colonize our skin and can potentially lead to an infection of the skin are types of Papilloma viridae, Polyoma viridae, and Cercoviridae. And the example that we'll talk about are papillomas or warts, and this is caused by human papillomavirus or HPV. And there are many types of HPV. There are over 100 types of HPV. Not all of them cause any type of medical or health problem, but there are other types that are related to different areas of the body. There are certain types of HPV that are related to cancers, like cervical cancer. But in all of these cases, it's human papillomavirus because of the relationship with forming papillomas or warts. And this is going to be transmitted via direct contact. And like I just said, there are many types of HPV that lead to different presentations based on where these warts or papillomas are occurring on the body. So there can be common warts, there can be plantar warts that occur on your feet specifically. And these warts that are visible and exposed can commonly just be treated and removed with liquid nitrogen. cutting them off with laser treatments, with topical ointments. But like I said, there are many types of HPV, and some of them are related to cancers, many of them are harmless. But many types of them do just commonly colonize our skin and are part of our normal microbiota. If you're interested in some other viral infections of the skin, there's this chart here. The only one that I'm specifically going to ask you about on the exam, though, are papillomas and types of HPV. Next up, we have fungal infections of the skin. And like we've seen kind of again and again, most often these infections are going to be opportunistic infections because fungi can be part of our normal microbiota. So if you're immunocompromised or you have some kind of cut or opening through your first line of defense, the fungi that's part of your normal microbiota can enter and proliferate and lead to a potential infection and disease. Primarily, fungal infections are going to be caused by moister environments. So that is parts of your body that are more moist, like sweaty shoes, leading to athlete's foot, and then places like community showers are also very common to transmit these fungal infections from one person to another. Fungal infections are also called mycoses, so just know that those are interchangeable. Mycoses also means fungal infections. Our specific example for fungal infections of the skin, or mycoses of the skin, is Tineas. So this is a group of mycoses caused by dermatophytes. And a dermatophyte is a type of fungal mold that uses keratin for growth. And think about this for a second. Where is keratin? It's in our skin. It's in that topmost layer of keratinized dead skin cells in our epidermis. So that is where these Tineas, or ringworms as they're also called, are going to grow and proliferate, and they're going to feed off of that keratinized dead skin on the surface of our skin. And depending on where this infection occurs, it's called different things. So Tinea corporis, for example, is ringworm on your body. Often you'll just hear it in terms of ringworm, but athlete's foot is caused by the same type of fungi that's causing ringworm. It's just called athlete's foot whenever it's happening on your feet. And something to keep in mind, though, it's called ringworm, but is this infection caused by a worm? No, it's caused by a fungus. It's caused by dermatophytic fungus that feeds off of keratin, so it's not caused by a worm. Also, these dermatophytes, or Tineas, or ringworm, so this type of fungi is commonly found in the environment and the soil. So it can kind of hang out on your skin, it can hang out on people, but it's also very common just in our environment. And therefore it can be very easily transferred to humans, or it can easily be transferred to humans from the environment or from contact with animals. So ringworm is a common zoonotic disease that can be transferred from animals to humans. And then also, there are lots of types of antifungal treatments that are effective against Tineas. You can just get this over the counter as a cream to put directly on the infection on your skin, and they're commonly termed azoles. So they have the ending azoles, azoles, for treating fungal infections, like miconazole. Again, a chart with some other examples of mycoses of the skin, or fungal infections of the skin. There are some very interesting ones here, but the only one I'm going to specifically be asking you about are Tineas, or ringworm. Finally, we will talk about an example of a helminthic infection of the skin. And remember, helminths are parasitic worms. So now we really are talking about worms. Ringworm, not caused by a worm. Helminthic infections, yes, caused by a worm. Our example is going to be loiasis, and loiasis is caused by the helminth, or parasitic worm, called Loa loa, and this is also known as the African eyeworm. This disease is endemic to West and Central Africa, and endemic should look familiar from the epidemiology lecture. Endemic means that it is specific to these locations, West and Central Africa. It does not occur outside of those regions. And it doesn't occur outside of those regions because it has a specific way or a specific vector that it uses to transmit from person to person. And it's spread through an arthropod vector called the deer fly. So a deer fly ingests larvae from the blood meal of one infected person, and then it transmits that larvae. Next time it bites a person, it deposits that larvae and results in an infection. And the development of these adult worms over time can survive for years and years and years in the host after you've been infected, and they survive in your subcutaneous tissue and move around the body. One place that they can move around your body as they're moving through your subcutaneous tissue is to your eye. Therefore, it's called the African eye worm, where it gets its name from, because it can actually become visible as it migrates across the eye tissue. And this is often how this disease is diagnosed. So oftentimes people don't even know that they have these worms in their body until it's migrating across the eye. And then some worms can be surgically removed. So especially whenever it's moving across the eye, it can kind of be itchy. Obviously it can cause distress, and these worms can be removed. But by the point that you're seeing worms crossing the eye, you have so many worms that they can't all be surgically removed. There are too many worms in the body. And it can be treated with certain types of drugs and medication, but these drugs and medication lead to severe side effects for the host. Because remember, helminths, like us, are eukaryotes. It's really difficult to develop drugs that specifically target another eukaryote. So these drugs can be really harmful to humans. And therefore, people don't always get these drugs because of these severe side effects. These worms are actually not deadly. This is not a life-threatening disease, and you can live comfortably with these worms also hanging out in your body, as unfortunate and uncomfortable as that sounds. It is more dangerous and deadly to receive the medication than it is to live with these worms. And finally, this is a slide from the Center for Disease Control, the CDC, showing the life stages of Loa loa. And I just find this really interesting because I really like parasitology. So if you ever take a parasitology class, it's extremely fascinating to see how these parasites move from host to host and then have these intermediate vectors for getting from host to host. I'm not going to ask you about this slide on the test. I just thought you might find it interesting to see how these worms are transmitted and their different life stages.